So, guys, you have seen how different sort of lipids are metabolized, whether it's their synthesis or whether it's their degradation. It doesn't only have biological importance; it also have very much medical importance. So, let's before we move towards the metabolism of other sort of macromolecules, let's have a very final glimpse of what we have learned about the lipid metabolism. So, as you know that triglycerides are one of the most frequent, is a major category of or major type of lipids which are present in our body. These, as far as the synthesis is concerned, of course, their major building block are the two carbon compound acetyl CoA compounds. These acetyl CoA then give rise to the formation of a very important reaction, what we say melanoate CoA, and eventually this leads to the formation of from C2 to C15 or whatever amount number of carbon atoms uh, are present in the fatty acid. So here this is example of C16 fatty acyl CoA, palmitic acid, what we say, or palmitoyl CoA, and then this is the fatty acid part. And then of course we know that the triacylglycerides are composed of fatty acid as well as the glycerol. So they are then interacted by the glycerol to make the triacylglycerol. Once the triacylglycerol are formed, they have to be deposited in the what we say in the different tissues, specifically most of the time in the adipose tissues. But for that they have to be transported, and for that transportation we make use of the proteins. These proteins are known as lipoproteins, and you saw that they then in this way these lipid molecules move from through different lipoproteins like LDLs, VLDLs, and so on and so forth. And not only with lipid molecules but also some other like cholesterol, not only the triacylglycerides but also the protein molecules and also the cholesterol molecules and also the phospholipids move along with triacylglycerides in these lipoproteins, and then finally they are deposited most of the time in the adipose tissue where the lipids or triacylglycerides are stored for to be further or later used whenever energy requirements are to be fulfilled. And then there is the degradation process, and we almost the reverse of the synthesis process, that the carbon 16 atom compound like palmitic acid, it has to sequentially degrade it by 2 units each, and eventually you end up with acetyl CoA molecules at the very end. Now these acetyl CoA molecules can either be go into the Krebs cycle to give you the energy, and because of this, because for 16 carbon molecules, remember we can get lot of ATP because of we have around 14 carbon molecule, 14 acetyl CoA molecules, so which really gives you not sorry for 16 carbon molecule palmitic acid, we end up with around 7 acetyl CoA molecules, which gives you a lot of energy because each acetyl CoA will go into the Krebs cycle. But also then if there is excessive formation of acetyl CoA because there is excessive degradation of the fatty acids, it leads to the formation of ketone bodies like like acetoacetate or acetoacetate or hydroxy butyrate or the what we say hydroxybutyrate or the ketone. So in ketone in the form of acetone, and remember that acetone is a metabolic dead end. So this is the triglycerides, how they are synthesized and how they can be used either in the Krebs cycle as acetyl CoA or as ketone bodies. Then we also talk about the phospholipids. We talk about two major phospholipid molecules or categories. One, the glycerophospholipids, and then the sphingophospholipids. The example of sphingophospholipids, of course, was sphingomyelin. Sphingomyelin under the action of sphingomyelinase give rise to sphingosine and the fatty acids. And any any problem, any failure in this degradation of the sphingomyelin can have really neurodegenerative related problems like Niemann-Pick disease. Similarly, then glycerophospholipids are degraded by phospholipases, giving rise to glycerol, fatty acid, phosphates, and alcohols. And these are then further used in different other metabolic pathways. And then we also saw the icocyanides, 20 carbon compounds, which are main precursor of which is arachidonic acid. This gives rise to leukotrienes, prostaglandins, and thromboxanes. And they further on, under the action of lipoxygenases and cyclooxygenases, give rise to the formation of other derivative compounds of these. And then we had finally a very important lipid molecule, the cholesterol, that it is a its synthesis is quite a complex pathway, but it also shows that its importance, because a lot of energy is required to synthesize cholesterol, but it also shows the importance of this molecule. The As far as the formation is concerned, 3-acetyl CoA give rise to HMG CoA, which gives rise to the formation of a very important compound, a rate limiting reaction. This is the mevalonic acid. And from mevalonic acid, and the mevalonic acid, from mevalonic acid onward, you have lots of condensation reactions and modification reactions, which ultimately at the end of the day give you a 27 compound cholesterol, a very energy intensive pathway. And as with any other pathway, this pathway also have many regulatory steps, but one of the most important regulatory thing is that it depends upon feedback mechanism. More cholesterol is there, lesser will be the synthesis, and vice versa. So finally there comes the, because we have to transport all types of lipids, and there is a dilemma, the lipids cannot move easily, conveniently through blood, because there is lipids are most of the time hydrophobic in nature. That's why they have to be carried by the proteins known as lipoproteins. There are HDLs, there are VLDLs, there are LDLs, and there are chylomicrons. All of those help in the transportation of proteins, and any defect in this transportation have some very important clinical consequences. One of these is, of course, the cardiovascular disease. So this is showing the metabolism of the specifically the triacylglycerols. And now here is the dihydroxyacetone phosphate, which gives rise to the formation of glycerol. It comes from the metabolism of glucose, which we in the glycolysis. Then you can also see that there are different amino acids that, like leucine, like tyrosine, which gives rise to the formation of acetoacetate, a very important ketone body. So finally, this is the acetyl CoA. You know, this is one of the most important component as far as the metabolism of lipid is concerned. This acetyl CoA also is a very important part of the TCA cycle, because it is the primary precursory molecule for the TCA, for the start of the TCA. So in this way, you can see that how the lipid metabolism is linked with the glyoxylate cycle, Krebs cycle, and of course with the protein synthesis and protein metabolism. It shows this importance of the lipid metabolism.